Thanks again for joining everybody. Today is March 19th. We've got a demo today from Quinn Wai Wong, who's at Oregon Health and Science University, who's going to be talking about variant analysis with the VRS Toolkit. I'm so excited about the demo, and then we can open it up to general Q&A beyond the demo. And then just a quick plug, we're also running our State of the AnVIL community poll. So this is an opportunity for you to share how you use AnVIL, kind of let us know what your thoughts are, and that helps us to improve the platform for users, help us to understand our user base a little bit better. So there are a couple of incentives with gift cards. So if you're interested, I'll put the link in the chat, and please take a couple of minutes to fill that out if you can. So with that, I'll hand it off to Quinn Wai, and we can dive into the demo.
Hey all, good morning. Hope everyone's doing well. Let me get my screen share started. All right, can everyone see that? Okay, great. So hey, thanks for the introduction, Natalie. Today I'm going to be talking and giving a little demo about VRS and how you can make use of the VRS AnVIL toolkit that we've created. I'm joined by Alex Wagner, who will cover some of the intimate details of VRS, and from there I'll sort of dive into the demo and how that applies to AnVIL. So with that, I'll probably hand it off to Alex to kick us off.
Hi. Yeah, great. So I think I recognize a lot of the faces on this call, but for those of you that don't know me, I'm Dr. Alex Wagner. I'm a principal investigator at Nationwide Children's Hospital in Columbus, Ohio. And I'm one of the primary specification maintainers for the GA4GH variant representation specification. And so when Quinn Wai mentioned to me he was giving this talk, I gave him some background material, so I'd be happy to join in and help out wherever useful. So I'm here to give some introduction, but also to stick around for Q&A if there are any questions about the spec itself or anything like that. All right. So to give just a bit of an introduction about where we're coming from. So my research, my area of focus has been on addressing the variant interpretation bottleneck. Seen here in the middle, we have our genomics researcher or variant scientist who's trying to pull together a bunch of different information from these different knowledge bases that exist over in the top left portion of this figure. And this it requires a lot of knowledge about the intricacies of each resource and about how to access that data and how to use it. Of course, from my understanding is we have a lot of bioinformaticians on this call, people that understand the challenges of data munging and trying to make different resources work the same way. And of course, there are like these efforts that exist to create like these harmonized data warehouses that do pull from multiple such upstream knowledge bases. That's the bottom left side of this figure. And these types of data warehouses are useful, but you still end up with several of them, and there's still kind of this multi-interface problem for the analyst. So what we're trying to do in the Global Alliance for Genomics and Health, which is a data standards organization, is build standards for how we represent and share genomic knowledge to make it easier for such knowledge bases to communicate interoperably at the data level. And this allows that same data to be used in a variety of different applications, not just for precision medicine and these clinical portals, but also, for genomics researchers, for education, and for AI-driven discovery. And so the goal of work like VRS is to help us achieve that. All right, next slide, please. So what is VRS? So the Variation Representation Specification, VRS, or VRS for short, is at its core a terminology and information model. We spent a lot of time thinking about what are the various individual building blocks that constitute variants. And how can we, put those together in ways where each one individually always means the same precise thing, and that in aggregate they mean a quite precise thing, such as a substitution variant or an insertion or deletion, a structural variant, etc., using all of the same vocabulary and that same information model. So we spent a lot of time teasing this apart. This was the labor of many people in this community over many years. And then from there, we built around it kind of a JSON schema protocol and some conventions. But that same terminology information model can be used in other exchange formats and with other conventions. But then on top of those standardized ones we used in GA4GH, we built a reference implementation. Now, over on the right side of the screen, what this allows us to achieve are some really useful characteristics of Verse. And first, Verse is interoperable across formats, so it works well with JSON, but it also works in, say, protobuf and in XML. It's also self-descriptive, right? That modular nature where we have these different components allows us to take this building block approach to building variation in a way that is understandable by machines and describes, and there are no sort of like assumptions or inferences built in, as you see in other common variant specifications where you have a more constrained space. And this is important because this allows us to have that evidence associated with variants be federated, where people talk about variants in different contexts with different meanings, and you can't make those sort of generalized assumptions about, kind of the one structure for your one data set, because in this space, there are many data sets with many structures. All right, and then finally, Verse allows for some global consistency and decentralization. So by virtue of the features of the specification, we're able to create these globally unique computed identifiers, which we'll talk about a bit later, which is a nice way to get resources. I'm sorry, was there a question? All right, maybe somebody was just had their mic off for a second there. Yes. So, anyways, that globally consistent and decentralized property of Verse allows us to have this without prior coordination, these different variants that can have the same computed ID. And we'll talk about that, as I said, again in a bit. So next slide. Now, some of the underlying conventions of Verse are interresidue coordinates and full-length justification. So we'll start with the interresidue coordinates. Now, this is an important distinction from other common formats like VCF and HGVS, which I'm sure everyone here is familiar with, where while those use a residue-based format where you provide a number and that indicates which residue in a sequence you're talking about, Verse follows the same conventions as something like BED or NCBI's SPEEDY format and uses interresidue coordinates. And there's an important, from a knowledge search and a knowledge aggregation perspective, there are certain useful properties of this. One of these is that the coordinates are no longer tied to the variant event type that they're describing. And to illustrate this, you can see these two examples here where we have an insertion that occurs between an A and a G as white characters shown in this sequence, or a deletion or substitution of that A or G in a sequence. Now, if you are in a residue-based system and you say, I want to insert between that A and that G, you'd say the insertion's between 6 and 7. And so your coordinates 6, 7 are exclusive, right? You're not actually talking about those residues, you're talking about the space in between them. Whereas if you're talking about deleting those two residues or substituting them, you again say 6, 7. But in this case, it's inclusive, and you're saying you're including that 6 and the 7. So what the coordinates mean varies depending on what type of variant is being described. And so you can no longer get the sort of generalizable notion of the coordinates of a variant. Similarly, but in contrast, the interresidue system allows you to say, for example, in the space 6 to 6, which is the space in between that A and G, we have an insertion, or from spaces 5 to 7, we're including that A and that G, and we don't need to worry about what that 5 to 7 or what that 6 to 6 means. It always means the same thing, regardless of the variant. All right, next slide. The other important convention that we use is again not aligned with VCF or HGVS, and it has to do with how we represent insertions and deletions in repetitive regions. So here we have this string TCAG It has this repeating CAG motif within it. And if you were to insert a CAG somewhere arbitrarily into that sequence, we can only observe that end state, right? When we sequence it, we only see the end state. We don't know where that insertion actually occurred. And so what different variant standards do is they have different conventions for representing that change. So here, where we have this CAG insertion, in reality, let's say it's inserted right in the middle of the sequence, as we see here in the black text. Um, what will happen is with VCF, if you could advance one. Thank you. With VCF, what they'll do is they will say all the way to the left of this repeating region, we will add in that CAG. And then if you're dealing with HGVS, again, advance, we see that this gets added all the way to the 3' end. And I say 3' because, and it's actually like, 3' or C terminus, but the idea being that it's always at the end of the sequence, which may or may not actually align with the corresponding genomic alignment or genomic orientation. So, but in essence, they always put it at the other end. So it might not, and often doesn't, overlap with the VCF representation of the same variant. Now, what Verse does and what the NCBI Speedy does is have a fully justified representation. And what you do is you take that entire ambiguous region and you replace it with that same ambiguous region with the change included, either as a deletion or an insertion. And so what we're doing is we're getting rid of that over precision and that ambiguity of where to place that variant, and instead saying this is what we actually know. And this has some useful properties for search. Next slide, please. So you can imagine if you have an insertion and you're saying, I want to, query the particular range for where that insertion might be, as shown in this teal bar. If you have that insertion that we just discussed happening, you can see that, like, if you're searching across an HGVS representation data store, you're going to miss it when your query is on the left side of where that insertion should be, but the data representation, which is HGVS based, is on the right side. But you'll catch the Verse Speedy one and you'll catch the VCF one. Next slide. Conversely, if you're searching from the right in like the HGVS style property, you might catch the HGVS representation. You'll catch the Verse Speedy one, but then you'll miss the VCF representation of this event. Next slide. So the nice thing about using this fully justified normalization is that you're going to improve your sensitivity of finding those insertion events or deletion events by virtue of having this fully justified representation, because you're going to cover that entire coordinate range. Okay, next slide. So now we move on to verse variants and global identifiers. So one of the nice things that we wanted to build from this strict data model was the notion of we can use that alongside a standardized convention for creating a computed digest to build computed identifiers. And those identifiers can then be used across resources such that no matter what the name of your sequence is, the variant residing on the same sequence by virtue of its content is going to result in the same variant identifier. So resources can have the same variants without needing to coordinate on things like what is the name of my sequences. And shown here in panel B, we have multiple such HGVS descriptions, the protein, transcript, and genome level, where we have different names for these sequences, but the same change that occurs on them. And they all end up with the same computed identifier by virtue of having this approach. Okay, next slide. All right, so all that's theory and why we built it and what it's about, let's talk a little bit more nuts and bolts. So one of our early works applying VERSE at scale was in the Nomad resource. I assume most people here are familiar with Nomad, the genome aggregation database. But for those that aren't, just know it's huge. It has billions of variants. And so what we wanted to do is take a look at those variants, which are represented using the VCF conventions, and build into it support for verse conventions, that fully justified normalization and those computed IDs. So we used Verse Python, which is a reference implementation under the GA4GH, which relies on a couple of upstream repositories, Biocommons seq repo, Biocommons bioutils, shown here, for sequence access and normalization methods, respectively. And then Verse Python, which is this big GA4GH symbol in the middle with the Python symbol on the lower right, that it was taking in basically GVCFs that come out of the Nomad V4 genomes and exomes tables. It annotates them and then returns that annotated VCF for ingest back into those tables. So our pipeline, our tooling is built around VCF as a file format, but it worked well in this case for us to use that as an exchange format between kind of that Hail representation and the annotations we needed for verse. So we did this at scale. We did it for a few billion variants, and now this exists in the Nomad V4 resource natively, so you can query by verse IDs and by verse range. Okay, next. More recently, we've been moving this toolkit, this stack, over onto the AnVIL platform, of course, which is where we get to today. And we're doing this in the context of the Gregor Consortium work. So for the Gregor Consortium, we are working on building evidence statements derived from combined consortia data. And so we're going to take that full cohort, or we are taking that full cohort, and we're computing basically the frequency of different genotypes basically on a on request basis, so on the fly, and returning those using basically these verse indexes. So the notion of we have these the entire cohort with verse identifiers on it using these verse coordinates. We can search by that identifier and quickly retrieve kind of like all the different genotypes, calculate it, and build a standardized evidence packet that can then be used in downstream research and clinical interpretation workflows. Now, there's a lot going on in this slide. I tried to summarize it quickly in 2 or 3 sentences there, but in short, the key work that we're doing here are those teal boxes on the left, and that's what we're going to be talking about today. And then that Jupyter notebook, I think that's kind of what Quinn Wai is going to be showing us about, like in the center, which is how do we use some of these tools on the AnVIL platform to do things like variant translation, do search across different resources, and that sort of thing. All right, next slide. All right, so today we're going to be talking specifically about our verse annotator, which is a workflow we built that takes in all these different components, applies verse Python on AnVIL, and gives you that annotated VCF. And I think that's the last of my slides and where we start moving into Quinn Wai's presentation.
Yeah, appreciate the comprehensive description and context behind verse. So now is the part where we get to dive into AnVIL and sort of understand how this works within that context. So to get started with the demo, I just wanted to point out we're doing all of this on a Verse AnVIL workspace, which is public and accessible to the broader AnVIL community on Terra. And so everything we're going to do is going to start from there. I'm going to try and walk y'all through two main use cases, which is connecting verse through your data. We sort of preface that a little bit with the verse annotator, as well as using verse to query in an external resource. And then if we get a chance to, I'll also briefly show y'all how to use verse for phenotype aware variant queries or for dynamic cohort variant queries on AnVIL data projects. So I'm gonna turn it over to, or I'm gonna move this over to AnVIL now, particularly this Verse AnVIL workspace. So as I said, this is public. This is a public workspace. You should be able to access this right now after this demo. And to get started, what I did was make a copy of that workspace. And as you can see, it's got everything carried over. There are some example VCFs, which are some stubs of um, different 1,000 genomes VCFs, since we want to work with open access data, make that shareable and easy to get started. I've also imported some 1,000 genomes participant level data just as reference. We've got some analyses notebooks here, which I'll dive into later. And finally, we've got the verse annotator workflow. And I think the verse annotator workflow is where I'm going to get started with how to connect some of your current variant data to verse. And so what the verse annotator is, it's essentially a workflow that wraps some of the verse Python tooling that's been created. And the goal of the verse annotator is to make it simple and fast to easily take any VCF that has any sort of annotation, any sort of details in it, and quickly get for each of the variants in there a verse ID and add that to the info field in a VCF. The goal of that being that to work towards that interoperability and make it easy to interoperate for any data set, regardless of whether you're working with a small amount of data or if you're working, on Gregor with a lot of joint VCFs and many participants. So first I'm going to just sort of run you through the workflow and how I might run a VCF through this. So first of all, I want to figure out the input VCF that I want to annotate, that I want to add verse annotations to. So for the sake of this, let's choose this VCF here, and this compressed VCF as well. You can see from previous runs, I have already written an output VCF that is annotated. So I'm going to try and rerun this again to sort of demonstrate that point. From the inputs, the most important input, of course, and this is pre-populated for you, is the input VCF that you'd like to annotate. That gets pre-populated from you based off of what you selected before. An output VCF name, and a reference and a pointer to the reference data that we're going to take use of. In this case, what Verse makes use of is seek repo, which is just a collection of reference data that gets loaded on the fly and handled for you. There are other settings. I won't dive too deep into these now. These are all have default options for you to investigate. In the future, if you'd ever want to take a look more at what those options are, there's more details here for inputs and outputs. One more thing I want to highlight before launching this is that I sort of prefaced before that there are columns that already exist. So this is just mirroring a Terra data table that's on the workspace. If you want to write directly to that table and not have to worry about, adding any of the Google URIs that point to these VCFs in the future, all you have to do is double check, and this is again pre-populated for you. But make sure that this writes to an output VCF, that output VCF column and output VCF index. And that's super helpful because then jumping over real quick to the those data tables, you don't have to automatically write those for you. Just a quick tip and something that I feel like is helpful when running workflows. So real quick, I'll just take a write down what workflow I'm running. So running 1,000 genomes VCF for annotation. And let's get that launched. So you'll see that that's being queued and will be in progress. I'll spare you guys waiting for this workflow to complete and sort of give you a sense of what happens once that is completed. So real quick, I clicked into the submission ID to make sure that I can uh, sort of drill down into where my VCF is located. And I can click in. So I had previously selected two different VCFs. So I can take a look at those VCFs here. Um, Going deeper in, you can see that I've found the VCF here. And from there, you can access it by clicking on one of these URLs. It will download it for you, and you can access it locally. If you wanted to access it on the platform, um, you can do so by copying some of these URLs and just copying it to your workspace. So that's a little bit about how to get a workflow started and annotate verse. As you can see, it's pretty straightforward. Oh, wow, that was quick. All of this stuff gets populated for you. And so as you can see, all of that status gets tracked as well. And so you might be curious to think, okay, maybe I don't have a VCF annotated already. How can I plug and play and get started with verse um, on Terra just without that? And so in order to get started with that, let me close a couple tabs here. I wanted to show you a little bit about how you might be able to use Verse and make use of some of the toolkit when you maybe are still waiting, which happily we're not waiting for that yet. And I'll show you how to use that annotated VCF as well. But for now, let's say, oh man, I still want to work with Verse. How could I maybe query an external resource, making use of those Verse IDs? As a way to interoperate or to reference other variant information in an external resource. And so there's a couple setup steps, including similar setup steps that are that we do within the workflow, which is installing the toolkit and making sure your reference data is available to your workspace. Now, since it's a workflow, we can't automate some of that stuff for you. So some of that stuff is a one-time setup, and then you're ready to go. I've already done that setup, so we can dive straight into converting a variant and seeing how we might connect that up. So as you can sort of see here, we are grabbing the verse package and making use of that for a single in this first example, I'll just do how to translate a single variant. If you sort of know beforehand, okay, this is the variant I want to study. This is how I want to translate it. This is I want to check and see if there are any references or any matches in an external resource. I'm going to do that initially. Um, and so initially, what I'm doing here is importing some of the verse tooling, as well as importing verse AnVIL, which is the toolkit that I have that we've sort of prefaced before. From there, all the steps required to translate the variant ID, it's really straightforward. All we have to do in this case is to provide a reference in this to the Seek repo data. Actually, in this example, we don't even need to localize our Seek repo yet because we're making use of a remote instance of it. That's super helpful if we want to just get started, annotate or get convert a variant to a verse ID quickly. Then we make use of this allele translator object, which will translate that variant to a verse ID, making use of this Seek repo reference as well as the allele translator. And finally, that returns a allele object. What Alex mentioned before is that this is super helpful because it not only contains a digest that is a fixed length digest that's computed, it also contains details about the sequence itself non-ambiguously, as well as its coordinates and the actual alteration that had happened at that position. And so for the purposes of this, we want to grab out that verse ID. And so I've saved that. From there, we want to know if there are any study matches. In particular, we're working with MetaKB to sort of highlight and tie back into that idea of wanting to connect up different possibly data that might be siloed into more of a unified knowledge base. And MetaKB has done that by taking things like the Civic Molecular Almanac and others to into a singular knowledge base so that it's easily queryable via Verse ID, which we know to be interoperable, and we know we're going to get a response back that this is okay, this is a matching variant with matching evidence. And so when I run that, essentially this is all you need to query MetaKB. It's just this function call and the verse ID at hand. And from there, we're just extracting the study level data, just grabbing that first study that's a match and formatting it into a way such that we can almost think about it as a sentence, an evidence statement, where for this particular variant, with this original label, HGVS label, and allele type, supports predicting sensitivity to a particular therapy, in this case, carboplatin, if I pronounce that right. You have to pardon my biology pronunciation and knowledge here. Primarily work as a research engineer. And it's reported in this study with a particular reference here and the title. And so, there's a lots of things that get returned to us in that MetaKB response. But here are some other things. You can see details about that this came from Civic, and this is an evidence ID from evidence data from Civic. It concerns variant therapeutic response, and you can see the germline variant, that it's a germline variant, and a little bit of a description. And so just to, tie this back and show you that just a different way of showing you the Civic data, they have an awesome platform that you can sort of reference off of and take a look at if you wanted to see this more in a visualization format before diving into and processing that data and continuing to process that data on Terra itself. And so you might be thinking from here, okay, I've done one variant. How do I do this for multiple variants for a VCF, in fact? There's a sort of similar process that has to be done. We'll make use of that seq repo reference data and also try and understand more how to do that. And we'll understand how to do that in the following setup. So I've imported a couple more parts from the Verse AnVIL toolkit, as well as some other Python imports, and setting that VCF path. Now I'm using a VCF that is a couple of chromosome 1 variants from the 1,000 genomes. And in order to set this up, what we do is that part of the toolkit is, the goal of the toolkit is to make this easy for the user to be able to plug and play. And so what we took is this approach of sort of a workflow. You've got inputs that you want, particularly a VCF files that you want to parse, any dependencies that you want to point to that are specific to your workspace or your Jupyter environment. And there might be performance parameters that you speed up that you might want to do, including threading, caching, limiting maybe the number of rows to do for testing for particular VCFs, and normalization. And so this is sort of like setting up parameters to a workflow, sort of like how we demonstrated at the start. And so in order to do that, what we're doing is just making use of a template manifest and filling in anything that's sort of relevant to us. So particularly those VCF files and the MetaKB directory, making sure it's linked up properly and that we have the files that we need to get started. This is what a manifest looks like. It contains all of that configuration for you. And then from there, all we need to do is call this annotate all function to get the metrics and and determine if there are any matches to the MetaKB resource. So what that does is it reads in the contents of this manifest above, calculates any verse IDs that might be relevant to it, and checks if there are any MetaKB matches for that particular variant. And our goal is to sort of provide a little bit of progress, show you how fast these variants are being annotated. And you can see it's, about 400, 350 variants per second. Awesome. So that has outputted a metrics file for us. We can quickly open that and see at a glance how long this um, annotation slash evidence matching function took, as well as see for each file um, any matches to MetaKB that we achieved. So from there, let's dive into we'll do a little bit of data processing. I won't bore you too much with the details because I want to just show you how you can get from verse into other resources and into other external resources. So um, in this part, we're basically going to just parse some of this existing data that we got from the metrics file. It's most importantly checking that if there is a match, let's update this. Let's update this matches per file dictionary so that for each of the VCFs paths, we have its set of matches. And from there, we'll want to parse that again and just make sure that for each of those matches, um, we now grab the allele that's associated with it and map any of the specific details that were used to gather that allele. So the file path that it came from, the original format of the variant, as well as the original ID of that particular format, as well as the results that come up. And so you can see that we've gotten a match to Civic again, this variant therapeutic response, and we've got two studies that match to it. We've Another important thing to note is that it was just making use of that singular querying MetaKB function. And we've got a little bit of a description for each of them. If we wanted to dive into those those pieces of evidence, we could do so by jumping to the platform and the UI provided by Civic. And if we wanted to parse or even look more into or do further processing for that, we could take that whole JSON that we'd created from MetaKB and make use of anything that's there and form those evidence statements and so on. And so no notebook is complete without a little congratulations at the end that you made it through. So that's a little bit about querying external resources with Verse. I wanted to show how you could go from, I don't have, I've got some variants, but I don't know if I can use I haven't used Verse with it, to using Verse, to making that connection to an external resource. I know we're a little over time, but I did want to just preface a little bit some of the details that I was talking about before with creating allele frequencies and doing queries with dynamic cohorts. I know that might be of interest for some people, and I'll just briefly preface this and then maybe open it up for questions. And if you're more curious about this, I can dive even deeper into it. But essentially, we've been doing a lot of work with Gregor in particular and with AnVIL in general to try and formalize how can we, I have access to a particular data project and I want to know, I'm interested in these particular sets of variants. How can I very quickly get allele frequency counts, not just in a joint VCF, not just for all of the samples that are available or all of the subjects that are available, but only for a particular subsetted on a particular phenotypic property or some shared trait that's in common with a cohort. And so we've done a lot of work both to the standardization point in making this standardized so that not only are you returning an allele frequency, but are understanding where it comes from a particular VCF and how that cohort is defined, but also that we're able to also able to grab this and ensure that this is not only a simple object that is returned back to you in the form of a cohort allele frequency, but that we know how it was tracked. So we know that maybe this cohort was only for participants that were enrolled in the US. And, that preference is some part of this demonstration where we, since we don't have access to, maybe Gregor outhand to work with a 1,000 genomes data and filter down a cohort of 1,000 genomes data based off of maybe their country of enrollment or based off of other details that might be available to them. And so creating sort of a framework for that is part of the ongoing process that we're doing at the Toolkit. And just sort of wanted to leave that for y'all as a final thing. And so just to sort of tie it up, today we sort of talked about how to annotate your VCFs, get that started and connected with by means of a Terra workflow, how to run some example notebooks, making use of a Jupyter environment, connecting that data to external data sources by using the analyses tab, and particularly this time in a Jupyter notebook. And I sort of prefaced, but not happy to answer more questions about that, how you might be able to use for a given AnVIL project, dynamic cohort usage and getting allele frequencies for it. And so just to wrap things up, thanks so much. You can find us on GitHub. We're very active on our repos, GitHub issues, discussions. And so if you have any concerns, questions, contributions even that you want to make to some of these and discuss with us. This is our GKS AnVIL organization where all of our repos are housed. And yeah, that's essentially it. Thanks so much. Sorry for going a little over time. Happy to open it up to questions, and I guess I'll pass it back to you, Natalie.
Thanks so much, Quinn Wai.